Hello friends, welcome to eStudent. In this video, we will be discussing Iit Jam 2018 question paper, and in the previous video, we have discussed the section A, and in this video, we will be discussing the section B and the section C, and the link for the section A is given in the description box. Okay, so now let us start discussing the section B. First question, antibody binds to antigen in a solution through. Okay, so first thing we need to know that antigen and antibody will always interact with each other via a non-covalent bond. Okay, so they will be forming the non-covalent bond. So here the options is ionic interaction, hydrogen bonds, van der Waals interaction and hydrophobic interaction. So we know all of these A, B and C, D are the non-covalent bonds. So here four of the options are going to be the correct option. Section B is a multiple choice question, so more than one answer can be the correct. So here A, B and C, D are the correct options. Next question. Plasmid mediated antibiotic resistance in bacteria are acquired by. So what is plasmid? Plasmid is an extra chromosomal. Other than genome, bacteria other the chromosomes, bacteria also contains extra chromosomal genetic material, that is the plasmid. Okay, so they are having resistant to certain antibiotics. So resistant to that antibiotics in plasmid has been acquired by what? So hydrolysis by beta lactamase, penicillin resistance, yes, they acquire the penicillin resistance by hydrolysis. So this is the correct option. Expression of amino glycosidase modifying enzyme, kanamycin resistance, yes, they are also having the kanamycin resistance. Okay, the third option is mutation in DNA gyrase. So we know DNA gyrase is present in the genomic DNA, not in the plasmid. Plasmid is an extra chromosomal genetic material, but DNA gyrase acts upon the chromosomal DNA. So this is not the correct option. Overproduction of dihydrofolate reductase. So this is very important in the production of amino acids and all, which is again present in the um, uh, chromosomal DNA, but not in the plasmid DNA. Okay, so this is also the wrong option. So A and B are the correct options. So the next question, which of the following statements are correct for G protein coupled receptor mediated signaling? GPCRs contain 7 membrane spanning regions. So this is the membrane. Here 1, 2, 3, 4, 5, 6, 7. So yes, GPCRs contain 7 membrane spanning regions. So that is why they are also known as serpentine. Okay, so they are also called as serpentine receptor. GPCRs are linked to heterodimeric G protein consisting of alpha, beta, gamma subunits. Yes, this is also the correct option because here it will be linked to a heterotrimeric protein which is having alpha, beta and gamma subunits. In the absence of GPCR interacting ligand, the alpha subunit of G protein is bound to GTP and complex with beta gamma subunit. So in the absence of ligand, they are actually saying, so whenever there is no ligand, there it is bound with the GDP. But whenever the ligand comes and bind here to the receptor, this GDP is exchanged for the GTP. So this is removed from here and GTP is bound here. So what here in the question is given? Absence of ligand, alpha subunit of G protein is bound to GTP. No, here there is no ligand, so there will be no GTP. Here it will be having GDP. So this is the wrong option. In the presence of GPCR interacting ligand, that means ligand has come here. GTP is displaced from alpha subunit of G protein by GDP. No. See, whenever there is an income of ligand here, whenever the ligand binds here, what happens is this GDP is removed and the GTP is added. But what it is giving here, that GTP is displaced. No, GDP is displaced. So this is also the wrong option. So coming to the next question. Glucose is incubated with enzymes of glycolytic pathway except pyruvate kinase. So here we are taking the glucose. It is incubated with glycolytic pathway. Basically it will undergo the glycolysis except pyruvate kinase is not given. Okay, which will convert the phosphoenol pyruvate into um, uh, pyruvate kinase. Okay. Um, gamma phosphorus 32 ATP and unlabeled inorganic phosphate. Which of the following products are formed? Okay, so here they have given unlabeled inorganic phosphate and labeled phosphate in ATP source. Okay, so we know that first step in the glycolysis is the formation of glucose 6 phosphate. So here it will be using ATP. Here our ATP is labeled. Okay. So glucose plus ATP will be giving glucose 6 phosphate, and here this is going to be the labeled phosphate. So this is the A is the correct option. Fructose 1,6 bisphosphate. Yes, here also the phosphates is provided by ATP only. Here this is this phosphate, this one, and here this phosphate is again provided by a new ATP molecule. So here this ATP which is labeled will be giving the phosphate. Coming to the third option here, 1,3-bisphosphoglycerate. Here in this step, one phosphate group is given by the inorganic phosphate. Okay, inorganic phosphate. So in this step, the phosphate will not be labeled by the ATP, but it is labeled by the inorganic phosphate. But what we are saying that here they are given unlabeled organic phosphate, but here it is showing that it is having a labeled phosphate at the third carbon. So this is absolutely wrong because it should not be labeled here. Okay, coming to the fourth option, yes, this one, this phosphate is also provided by the ATP. So this is going to be the labeled phosphate here. So D is also the correct option. Now coming to the next question. In a double-stranded DNA, which one of the following ratios is always equal to 1? Okay, so we know according to the Chargaff rule that A is always equal to T and G is always equals to C. Adenine, thymine, guanine and cytosine. Okay, so we can, by this we can also say that A plus G is equal to T plus C, or we can also say that A plus C is equals to G plus T. Right, we can say this. So from this we can also say that from this we can say A plus G by T plus C is equal to 1, and here we can say that A plus C by G plus T, sorry, here G plus T is equal to 1. Okay, so we can write it like this, right? A plus G, sorry, we can write it like this: A plus C by G plus T is equals to G plus T by A plus C. We are just writing it this here and writing this here. Okay, so here you see A plus T by G plus C. Are we getting this combination here? So no, this is the wrong option. A plus G by T plus C. No, this is also not right. A by G. No. Here we are getting G plus T by A plus C. So yes, D is the correct option. Hope you have understood how we have solved this. A plus T, A is equals to C and G is equals to C. So we can write it as A plus G and T plus C here. Okay. Or we can also write A plus C here, like A is equals to T and G is equals to C. So we can also write A plus C is equals to G plus T. Okay, so after this we can write A plus C by G plus T is equals to 1, and then we are just changing these positions. G plus T by A plus C is equals to 1. Okay, so D is the correct option here. Next, the total number of genetically different types of gametes that will be produced by heterozygous plant carrying the genotypes AA, BB and CC. So this is a section C. Yeah, so here they are asking heterozygous plant carrying the genotypes AA, BB, CC. So how many genetically different types of gametes will be produced by this? So for these type of questions, we'll be having a general formula, that is 2 to the power n, where n will be equal to the number of heterozygous pairs in genotype. Okay, the number of heterozygous pairs in the genotype. So how many heterozygous we are having here? B and B, C and C. So they are two heterozygous. So 2 to the power n, that is 2 to the power 2, that is going to be 4. So here correct so section C is a numerical type questions we are going to have. So 4 is the correct option. Next question. A healthy individual has cardiac output of 5.5 liter and the heart rate of 72 beats per minute. The stroke volume of the individual is dash ml. So the question is, in question we are having the cardiac output, that is CD, and the heart rate HR, and the stroke volume. So we need to find out the stroke volume. Okay. So we know that the cardiac output is always equal to stroke volume into heart rate. Okay, so this is the actually the question from animal physiology. So this is, we can't do anything, just but we have to remember the formula here. This formula is something which has to be remembered. Okay, so here they are asking stroke volume in ml. So whatever is given, cardiac output is given in liters. So we have to convert this cardiac output into the ml first. Okay, so what is given, cardiac output is 5.5 liter, which we convert into ml, we get 5500 ml, that is 5500 ml, which will be equal to stroke volume into what is the heart rate given. Heart rate is 72. So the stroke stroke volume will be equal to 5500 divided by 72, which will be equal to 76.4. Okay, so this is going to be 76.4 ml. So hope you have understood this. Yeah. Coming to the next question. Both strands of a DNA molecule are labeled with radioactive thymidine and are allowed to duplicate in an environment containing non-radioactive thymidine. The number of DNA molecules that will contain radioactive thymine after 3 duplication is. So what they are saying, there is a DNA molecule. Okay, so there is a DNA molecule and both the strands are labeled. Okay, so I'm denoting these balls like they are labeled here. Okay, so both the strands are labeled with thymidine and allow duplicate in an environment contained. So they are duplicated in non-radioactive thymidine. Okay, after 3 generation, that is 3 duplication, how many radioactive thymine we are going to get? They're asking. Okay, so let's see what happens after the first generation. So first generation we'll see. Okay, first duplication. So one here we are going to have the one from here. This is going to duplicate, and we'll have one radioactive DNA, and the other here we are going to have 2 radioactive DNA here. Then second duplication. Let's see what happens. Here this will be the radioactive, and this one when duplicate, this is non-radioactive. This is again radioactive, and this is going to be non-radioactive. Now let's see what happens in the third duplication. Now this is going to be the radioactive, non-radioactive, 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 radioactive. Sorry, this is going to be non-radioactive, non-radioactive. Okay, so see, I'm just duplicating these ones here. See, this one is duplicated, again this one is duplicated here. So we are getting only 2 DNA molecules which are radio labeled after 3 duplications. So here we are going to get 2 DNA molecules. Okay, yeah. Coming to the next question. The number of cycles required for a complete degradation of palmitic acid by beta oxidation is. So, palmitic acid is 16 carbon fatty acid. Okay, so in each cycle, one acetyl CoA is removed from the palmitic acid. So, one cycle minus one acetyl CoA is removed, which is 1 acetyl CoA is equal to 2 carbon. Okay, so from one cycle 2 carbon is removed. So 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16. So this is a 16 carbon palmitic acid. So in the so this is 1 carbon and this is 2 carbon, 4, 5, 6, 7, 8, 9, 10, 11, 12. Okay, so in the each cycle, just 2 carbons are removed. So 1 acetyl CoA is removed. So 1 acetyl CoA is equals to 2 carbons. So here we are removing the 2 carbons here. So here again 2 these things carbons are removed. From here 2 carbons, 2 carbon, 2 carbon. So in the each cycle we are removing the 2 carbon here. Yeah, here 2 carbon. Yes, so yeah, I'm so sorry here. Here they'll be it will be not able to break here because here no breaking because they are 3 carbon here. Okay, so it will here it will not be able to break. Yeah, so it will be breaking with 1 2 3 4 5 6 7 cycles here. So 7 cycles. 7. Number of cycles are going to be the 7 cycles. Coming to the next question. A 100 nucleotide long single strand poly A is synthesized from adenosine monophosphate at physiological pH. Atomic mass of C12, carbon is 12, hydrogen is 1, oxygen is 16, phosphorus is 31. At physiological pH, molecular mass of AMP is 345. The molecular mass of resulting poly A at physiological pH is. So what they are saying is that there is a formation of, like this is the poly A here, like this they are forming 100 poly A. So this is one and this is like 100 poly A tails are forming. So they will be forming the phosphodiester bond. Okay, they'll be forming a phosphodiester bond. So in every phosphodiester bond formation, there is a release of, there is a minus 1 H2O in every bond formation. So here we see that there will be 1 to like 99 phosphodiester bonds forming here. Okay. So the molecular mass of AMP is given is 345. Okay, 345. So how many bonds, how many AMPs are present? There are 100 AMPs present. Okay, so minus how many water we are having? We will water will be removed. That will be 99 water molecules will be removed. See, okay, because see there are 99 bonds forming, so 99 H2O will be removed. Okay, and the what is the molecular mass of water here? See, here it is 1 and here it is 16. So 2H we are having 2 into 1 plus 16, which is going to be 18. So 18. Okay, so here we will be getting 34500 minus 99 into 18 is going to be 1782, which will be equal to 32718. Okay, this is going to be 32178. Yeah, coming to the next question. The color blind woman marries a normal man. The chance of their boy getting a child will be color blind is. So color blindness is a X-linked recessive disorder. Okay, this is the X-linked recessive disorder, which is like HCN and HC. These individuals are going to get affected. They will be actually having the color blindness. So this color blind in women, this is happening in the case of women. Okay, they are mated with XY normal man here. Okay, so here it is going to produce like HC here, H and Y here. Okay, let's draw the Punnett square. Yeah, so we will be getting HCX CY CY. Here these both are the carriers. Here these both are going to be color blind, because this one here are the color blind because HC is the dominant one here. Hope you have understood. Thank you.